The spleen is the largest single organ in the lymphatic system and is typical of vertebrates, as is evidenced here by the images of the spleen in the shark, frog, chicken, opossum, pig, monkey, and the model of the human spleen. There are two types of tissue in the spleen, what is referred to as the red pulp and the white pulp. These are evident under the microscope, as can be evidenced here. The red pulp has more of a reddish or pinkish tinge because of the red blood cells there, and the white pulp has been stained more of a purple because of the stain used to stain the nuclei of the white blood cells. The major role of the red pulp of the spleen is to perform phagocytosis of old, worn-out red blood cells. Since red blood cells lack nuclei, they cannot repair themselves, and so after about 120 days, they may reach the end of their lives. They could burst in circulation, and this would be a problem given the grand number of red blood cells. The debris which would be created if they burst in circulation would potentially clog blood vessels. And so therefore, in the red pulp of the spleen, old red blood cells, which are less flexible, are identified by macrophages, which then perform phagocytosis and break them down. The hemoglobin is processed and recycled. The globin protein portion is broken down to amino acids. The iron is stored and recycled, and the heme is converted to bilirubin, which will be further processed in the liver. The red pulp is also a site where old platelets can undergo phagocytosis so that they do not burst and release clotting factors in circulation. And also platelets can be stored here so that they can be released in larger quantities following an injury. In fetuses, this spleen has a role in the production of red blood cells in erythropoiesis, but this is not a function performed by the adult spleen. The white pulp of the spleen is composed of reticular tissue in which a variety of white blood cells can then aggregate on reticular fibers and look for infectious and potentially dangerous elements in the blood. So infectious organisms can be identified, in addition to foreign matter and debris for phagocytosis. Lymphocytes, after identifying foreign organisms in the blood, can produce antibodies, and the white pulp of the spleen is important for the storage of macrophages. About half of the body's macrophages can be stored here and released given the appropriate stimuli. And so the spleen has important roles as filtering blood and looking for infections and foreign material in the blood.